Now, another thing that has, I think, really been striking from the genetic analysis of tumors, both in a classical genetic sense and also in a molecular genetic sense, is that different genes are turned on in different tumors. I think in 1971, when Richard Nixon announced the war on cancer, scientists as well as the public were very optimistic that we were going to identify a single gene or a handful of genes, or a single cause or a handful of causes for cancer, and that we would then be able to identify some sort of a silver bullet that would allow us to eliminate this disease. We now really appreciate that cancer is hundreds of diseases. It's a much more complex challenge. I've already given you an example of several cancer mutations that can cause different kinds of cancers, and we now have the capacity to actually look at all 30,000 human genes for their expression simultaneously using microarray strategies. And when we do this in this image here, we're looking at 1,800 genes running from the top to the bottom of this panel, and 140 or so different types of tumors running from left to right. And what you can see when you analyze this, with the green genes being green spots representing genes that are turned off and the red spots representing genes that are turned on, is that these patterns of gene expression actually segregate into groups. And in many times you can see that, for example, here all of these prostate cancer samples have very, very similar patterns of expression. And that but on the other hand, if you look down here in breast cancers, you can see that even though these in the box here are all breast cancers, genes expressed in breast cancers, you can see that they're not all green and they're not all red, illustrating that there's a great deal of heterogeneity even within a particular type of tumor. And this is really one of the major challenges, I think, for cancer biologists and clinical care providers that we're facing currently. It's very common for two women with breast cancer to have the same diagnosis, the same treatment, and have very, very different outcomes. One person may succumb to their cancer within a few months, another may live for 10 years. And we really now can appreciate, because of this kind of detailed molecular understanding, molecular fingerprinting analysis, that we really need to have a sort of a more high resolution view of the tumor itself in order to define the most appropriate treatment for that tumor. So I think a really major revolution in our thinking about cancer biology is this appreciation of the diversity both of tumor populations and of patient populations. And this has really come specifically through advances in genetics, molecular genetics, and the approaches that I've described today. We used to think that all tumors were the same and a tumor in any individual context was the same. We now appreciate that there are many, many differences in tumors. Even tumors of the same tissue type can be extremely different in terms of their properties, and therefore are going to be extremely different in terms of their responses to therapeutic intervention. And we also appreciate that the tumors within the context of an individual, even the same type of tumor within the context of a different individual, can be very different as well. And we've had a presentation in this series from Brian Drucker which addresses that exact issue. So I think that, again, this revolution in molecular genetics that we've seen occurring just very recently really gives us increased genetic understanding of the causation of cancer, makes us appreciate how diverse the mechanisms are that can cause cancer, but gives us a lot of hope that with that information we can be empowered to use new strategies to address this really major health issue. We can, as I've described, begin to identify individuals who are at high risk for developing certain types of cancers, particularly in the inherited forms of cancers, but maybe over time the sporadic ones as well, and then with that predictive power, develop guidelines for more aggressive screening and early detection. And then the goal would be, of course, to then prevent those tumors from ever developing, or at least screen and catch them early before they're very dangerous. And really this all leads to this view of a more personalized approach, both in terms of identifying individuals at risk, as well as using molecular strategies for high-resolution diagnosis of tumors, so that the tumors can be matched in a very rigorous and appropriate way to therapeutic intervention.